Good morning, students. So today we are going to study about DNA replication. As you can see here, this is a double-stranded structure of DNA. Now let us just zoom here. Okay, if you see here, this is a ribose sugar, a phosphate group, and a nitrogen base. Okay, so these three, they make up the nucleotides, the basic building unit of a DNA. Okay, now if you see here, this is having 5 carbons, 1, 2, 3, 4, and the 5th carbon. Okay, so phosphate group can be attached to the 5th carbon as well as to the 3rd carbon. The nitrogen bases are always attached to the 1st carbon. Okay, so this is your nucleotide chain. Now if we see here in the beginning, the 5th carbon is free, nothing is attached, so this is 5' end. Okay, if we see this strand here, if we count from here, 1, 2, third carbon is free. So in this one, you are having 5th carbon free here, and this one you are having 3rd carbon free. So this is a 5' end and this is a 3' end. If we move down this strand, this one, so what you are seeing here, this 3rd carbon is free. 1, 2, 3, 3rd carbon is free. So the strand is going from 5' to 3'. Okay, same here. Here 3rd carbon is free, nothing is attached to this, so this is 3' end. If we go down this strand, what we are finding here, this 5th carbon, CH2, 5th carbon is free, nothing is attached. So this is from 3' end to 5' end. Okay, and again another thing, if you see here, this, the sugar and the phosphate group is forming the backbone. Okay, the center is being taken by the nitrogen bases here. This is the center of the DNA molecule. Okay, now if you see here, the nitrogen bases, this is adenine and this is thymine. Adenine and guanine, they belong to purines. Okay, purines are having double ring, fused rings, while pyrimidines, thymine belongs to pyrimidines, they are having a single ring. As you can see here, this is a single ring and this one is a double ring. A and T are joined together with the help of 2 hydrogen bonds. Can you here see 2 hydrogen bonds here? Okay. While guanine and cytosine is a pyrimidine, single ring, and guanine is a purine. They are joined by 3 hydrogen bonds, 1, 2, and 3. So 3 hydrogen bonds. Okay. Now let us start with DNA replication. Okay. The DNA replication, it requires origin of replication, but for convenience sake, we are just going to we assume that we are starting with the origin of replication. So this is a very long DNA chain. We are having a more longer DNA chain in our body. It is like 2 meters long, so it needs to be opened up. And how will it get opened up? It will get opened up with the help of enzyme helicase. Okay, so here is the enzyme helicase, and what this enzyme is going to do, this enzyme is going to open up this strand. So here is the enzyme. Okay, and this enzyme is opening up this strand like this, pushing it away, giving it a fork-like structure. And how it does it? By breaking the hydrogen bonds between them. So you can see this. We are getting a fork-like structure. by breaking of hydrogen bonds till this. Okay, now we were having two strands here. One strand, if you see here, this is 5 dash to 3 dash. Here the 3 dash end is free. Okay, another strand that we are having here is 3 dash to the 5 dash end, where the 5 dash end is free. Okay, so the strand which is 3 dash to the 5 dash end, this one is known as the leading strand. Okay, why is this so? Because the DNA polymerase can move only in 5 dash to 3 dash direction. It cannot move in any other direction. As a result, this strand will become the leading strand and the opposite strand will become the lagging strand. Let me show you how this happens. So this is our replication fork. Okay. Now once the DNA has opened, the strands have been opened up, they need to be stabilized because it's a very long strand and they need to be stabilized. So for that we are having the proteins, single stranded proteins which will fix the strands in their places so that they do not recoil back or join back. Okay, so here we are having this can be taken as the single stranded protein which has just stabilized the two strands. Once the single stranded proteins have attached, still we require one more thing. We require DNA polymerase enzyme which will start this replication. Okay, then the DNA polymerase needs to know in which direction it needs to go. So because it has no idea which is the 5 dash and 3 dash end, for that an assistant is required. So assistant is basically formed by the primase enzyme. Primase will form what is known as RNA primer. Okay, RNA primer will synthesize 4 to 10 nucleotides long chain on the DNA strand. It is a DNA dependent enzyme. Okay, now because we are not having that long strand to show 4 to 10 12 nucleotides long RNA polymerase synthesis, so we will just use one nucleotide here. So this is your just take it as an RNA primer. It is showing the direction, so in this direction it has to go, so it will fix it here. So it is telling the DNA polymerase that you have to move in this direction. Okay, what about this strand? This strand is 5 dash to 3 dash. The DNA polymerase can only move from 5 dash to 3 dash, but because it is 5 dash to 3 dash, it cannot move in the same direction. So what will this do? The RNA primer will come and associate at this end here. Okay, and it will tell DNA that you have to move in this direction, which is now becoming 5 dash to 3 dash. Okay, so once the RNA primer is in its place, what will happen is the DNA polymerase, it will bring the selective nucleotides, okay, and put them into the position. So if we see here we are requiring this is adenine, so the complementary base pair will be thymine here. Okay, so we will put a thymine over here, or DNA polymerase will bring the thymine over here. Okay, so this is the DNA polymerase bringing the thymine and putting it into the position. Okay, now in the nucleotides these are triphosphates. The two phosphate bonds are broken, which also act as energy source for this polymerization reaction. Okay, so next one we are having, here is a thymine. This one is a thymine. So what we are going to have, we require a adenine. Complementary base is adenine. Okay, so this is also brought by the RNA polymerase. This is the RNA polymerase, and RNA polymerase is bringing it and fixing it in the position here. This one is adenine. like this, they are joined. The next one, we require a guanine. Okay, so again brought by RNA polymerase, bringing it and fixing it in the position here. The bond is forming and fixed like this. Okay, the next one, here we are having guanosine, this one here. Okay, so guanosine will come over there like this strand will keep on increasing. Okay, here this was our 5' to 3' end. The RNA primer was fixed here, so the strand will form like this. Okay, discontinuous one. Here we are having guanine, so complementary base will be cytosine, again brought by DNA polymerase and placed over here like this. Okay, then we are having, there is adenine A, so we require a thymine group. Another one brought over here, fixed like this, like this. Okay, then we are having here thymine, so we require an adenine. So here is this one like this. Okay, next we are again having a thymine, so again we require an adenine. So this one is fixed like this. Okay, furthermore it will keep on opening up with the helicase group. It will keep on opening up like this. Like this it will further open up. Okay, now here if you see here, the leading strand, this is the leading strand. Okay, leading strand will continue. The RNA primer need not be put again and again. It will only be in the first place. It will continue like this. But in this lagging strand, here the RNA primer was put to give the direction to DNA polymerase. Okay, here again another RNA primer will be put here, like this, and the strand will then continue. Okay, once everything is done, the RNA primers will be removed. Okay, particular nucleotides will be put and the strands will be joined together.